Q-qPCR software tutorial. Cycling analysis. In this video, we will show you how to complete cycling analysis for any run using the Q-qPCR software. Once a run is completed and the samples have been properly annotated, cycling analysis is straightforward. To start a new analysis, select from the following options: cycling, melt, high-resolution melt, absolute quantification, allelic discrimination, identifier, relative quantification. For this run, let's select the cycling analysis option to determine CQ values and reaction efficiency of each sample in a data set. Click on the plus button next to the desired analysis type in the navigator bar. Choose the target you want to look at. The analysis consists of the cycling analysis graph, calculated results, as well as the option to adjust the calculation parameters and samples displayed. The graphs display the normalized fluorescence in log scale by default, along with the automatically determined window of linearity within the gray area and the automatically determined cycle threshold as a red line. There are three graph types. Logarithmic y-axis displays data in the log scale and provides better visualization of the exponential region of the amplification curve. Linear y-axis displays the baseline corrected cycling data with the y-axis in linear scale. Derivatives displays the first and second derivative curves in parallel with either the linear or logarithmic charts. Cycling analysis parameters. Several cycling analysis parameter options are provided. Select the desired analysis method from the method drop-down list. LinReg-PCR is the default analysis method used by the software and sets the default baseline correction. Dynamic is the analysis method recommended when using hydrolysis probes. Fixed length is the most basic method. None allows you to view the raw data in cycling analysis. The method selected will depend on the chemistry type used for the assay. For this run, we used hydrolysis probes, and due to their accumulative signal, LinReg-PCR might not be a viable option. Instead, we choose the dynamic method. The switch shows significant improvement. Let's take a look at another target with a similar issue. Again, we need to apply the dynamic option for baseline correction. If we look at the first few cycles, we can see the baseline is not steady. We can ignore the first few cycles of the curve. The threshold is auto-calculated by the software. It can also be set manually by the user. In this example, we have a multiplex run with another dye, yellow. We use a fluorescence cutoff to exclude signal that might appear due to crosstalk from the other dye. If we remove this cutoff, we can see the crosstalk from the yellow dye. By ignoring samples which have signals less than 4% of the maximum signal detected, we can remove the crosstalk. The default value is set to 5%. Having adjusted the cycling analysis parameters, we can save the new values back into the assay file. Or we can reload the default parameters saved in the assay file. Results table. Results are presented in a tabulated format, which can display grouped replicate values. The mean and standard deviation of the CQ values are shown for each sample group. Individual sample data are reported in the columns. These include CQ values, reaction efficiency calculated from window of linearity, R² for curve and window of linearity, any additional information regarding the result. Collapse individual samples in a group by clicking on the diagram and selecting Collapse All. Or expand individual samples by selecting Expand All. Ungroup sample replicates to show individual samples in well numeric order by clicking on Sample Name. Sort the data in the column from lowest to highest value by clicking on the column heading, or highest to lowest by clicking again. Clear the sorting by right-clicking on the column heading used to sort samples and select Clear Sorting. Click on the diagram and select Sample Name to regroup replicates. Save the results table to CSV file format. Or copy the results table data to clipboard so that you can paste it into another third-party software. Remember to save your analysis. Files with an asterisk will require saving. In this video, we have shown you how to analyze a run using the cycling parameters to optimize the cycling analysis and get your results. For more information about cycling analysis, please contact the QuantaBio team.